What is RSV and why is it a serious threat to infants? RSV stands for respiratory syncytial virus, a common respiratory virus that causes mild cold-like symptoms in most people. However, for some infants, especially those born prematurely or with underlying health conditions, RSV can lead to severe complications such as pneumonia and bronchiolitis. Pneumonia is an infection of the lungs, and bronchiolitis is an inflammation of the smallest airways in the lungs. Both of these conditions can make it hard for infants to breathe and get enough oxygen. RSV is the leading cause of hospitalization and death among infants worldwide. According to the CDC, each year in the United States, an estimated 58,000 to 80,000 children younger than 5 years old are hospitalized due to RSV infection. Unfortunately, there is no specific treatment or vaccine available for RSV in infants. The only way to prevent RSV infection is to avoid exposure to the virus and practice good hygiene measures such as washing hands, covering coughs and sneezes, and avoiding close contact with sick people. RSV can be dangerous for some infants and young children, so it is important to recognize the symptoms and seek medical help if needed. Pfizer's maternal RSV vaccine, also known as RSV, is a promising candidate for preventing respiratory syncytial virus, RSV, infections in infants. RSV is a common and potentially serious respiratory virus that can cause pneumonia, bronchiolitis, and even death in young children. Currently, there is no licensed vaccine for RSV. RSV is a bivalent vaccine that contains two types of RSV prefusion F proteins, which are the part of the virus that attaches to and infects cells. The vaccine is designed to stimulate the immune system of pregnant women to produce antibodies that can be transferred to their babies through the placenta and breast milk. These antibodies can help protect the infants from RSV disease for the first 6 months of life, when they are most vulnerable. The vaccine has shown encouraging results in a phase 3 clinical trial called MATIS, which involved more than 7,000 pregnant women and their infants in 18 countries. The trial found that RSV was 81.8% effective in preventing medically attended severe RSV-associated lower respiratory tract illness (LRTI), and 57.1% effective in preventing medically attended RSV-associated lower respiratory tract illness (LRTI) in infants within 90 days after birth. The vaccine also reduced hospitalizations and intensive care unit admissions due to RSV by 78.4% and 90.6%, respectively. The safety and tolerability of the vaccine were also favorable, with no significant differences in adverse events between the vaccine and placebo groups in both mothers and infants. The most common side effects were injection site reactions, such as pain, redness, and swelling. Based on these results, Pfizer has submitted a Biologics License Application, BLA, to the U.S. Food and Drug Administration, FDA, for priority review of RSV for maternal immunization. The FDA has set an action date for August 2023. If approved, RSV would be the first vaccine for administration to pregnant individuals to help protect against the complications of RSV disease in infants from birth through 6 months. Pfizer has also filed a Marketing Authorization Application (MAA) to the European Medicines Agency (EMA) under accelerated assessment for both older adults and maternal immunization. A decision is expected in the second half of 2023. However, there are still some challenges and uncertainties that remain for RSV. One of them is the potential risk of preterm birth associated with the vaccine. In the MATIS trial, there was a slightly higher rate of preterm births before 37 weeks of gestation in the vaccine group than in the placebo group, 5.1% versus 4.5%. However, this difference was not statistically significant and there was no difference in preterm births before 34 weeks of gestation, 0.9% versus 0.9%. The researchers suggested that this finding could be due to chance or confounding factors, and that further studies are needed to confirm the safety of RSV in relation to preterm birth. Another challenge is the need for further studies to evaluate the long-term safety and efficacy of RSV in different populations and settings. For example, it is not clear how well the vaccine will work in low and middle-income countries, where RSV burden is higher and maternal immunity may be lower. It is also not clear how long the protection conferred by the vaccine will last and whether it will interfere with natural immunity or subsequent vaccination against RSV. In conclusion, Pfizer's maternal RSV vaccine is a breakthrough innovation that could save lives and prevent complications from RSV infection in infants. However, more research is needed to address some of the remaining questions and challenges before it can be widely implemented.